All right. So we're going to continue our exciting, in quotation marks, exploration of the cells by talking about the cell membrane or the plasma membrane. And we've said before that the plasma membrane, or the cell membrane, you can call it either one. Either one's work. Either term works. Separates the intracellular from basically the extracellular fluid. Okay, so if I have a cell here, and I have a cell over here, okay, and yes, the cells aren't perfect circles most of the time. I got the nucleus in here, okay. So the fluid on the inside, of the cell, that's intracellular fluid. That's intracellular. It's on the inside of the cell. And on the outside of the cell, every cell you have is bathed in this. And I'll make this, get a nice kind of purplish color. Out here, bathed in all these cells, wrapped around all these cells, along with other proteins and stuff like that, we have the extracellular fluid. And what keeps the intracellular fluid and the extracellular fluid from mixing together is the plasma membrane. Okay, so the extracellular fluid that surrounds all the cells, bathes them, and the intracellular fluid is the fluid on the inside of the cytosol. All right. Okay, so what does the plasma membrane do besides separates them? Okay, it's a physical barrier. It is. If you just want to get down to the brass tacks, it is a physical barrier. Okay, kind of like your skin is a physical barrier. Keeps the outside environment from actually getting inside of the skin itself. Okay. It controls how fast things enter and leaves the cell. So it is a transport regulator. Okay. It will monitor chemicals in the in the tissue. Monitors chemicals. And I'm just going to say the environment. Okay. And the last thing that it does is it will anchor cells. It is the anchor of cells to the connective tissue. Okay, it is the anchor to the connective tissue itself. Okay, so now connective tissue is something we're going to talk about when we talk about tissues themselves. But what the connective tissue is literally, they tell you pretty much, the name tells you exactly what it is. Connective tissues connect shit together. I mean, that's literally what they do. And so it's one of the 4 tissue types in the body. And basically what it is just a bunch of fibers. A lot of fibers with a lot of fluid if it's loose, and it's really densely packed fibers if it's dense connective tissue. And basically all the connective tissue really looks like is you've got cells, you got a couple of cells in here, and you've got these fibers that go along them, go along between these cells in haphazard directions. And these cells will actually be anchored to the fibers of the connective tissue. And it's usually proteins inside of the plasma membrane that allow them to be anchored there. So the plasma membrane allows for anchoring of tissues, of cells to the connective tissue themselves. right? So let's take a look at the general structure of the plasma membrane itself. So to start, the plasma membrane, so the structure, it is a phospholipid bilayer. Okay. That is the first thing and the most important thing right there. It is a phospholipid bilayer. And if you don't remember exactly what a phospholipid is, go back and look at the lipid lectures. So we go more in detail. But it is so bilayer means it is two, and they're just phospholipids. And it's arranged in such a way that the phosphate of this phospholipid bilayer faces the extracellular fluid and the intracellular fluid. So we can take a look. So we can just draw a bunch of circles here. So we'll just draw 4 circles there. And you got the lipid part, goes inside. And then on the other side, this is the bilayer. And you have the lipid part that goes inwards. And I drew this picture in the lipid lecture itself. So this is the basic structure. It's phospholipid bilayer. And it goes on in this direction, and it goes on completely in this direction. Okay. Embedded inside of the phospholipid bilayer, we have many integral and peripheral proteins with integral and peripheral proteins. Okay. So what an integral protein is, so let me erase some of these. Let me erase these arrows. So an integral protein is basically, it goes completely through the membrane. So I'll draw it in a green color. So if you have like a protein here, a protein that totally crosses the plasma membrane. So there's an extracellular, here's intracellular. If you have a protein that spans the entirety of the plasma membrane, so one side touches the extracellular fluid, the other side touches the intracellular fluid, that is called an integral protein. This is an integral protein. Okay. If you have a protein that is only on one side of the plasma membrane, let's say we're going to call this protein right here, and it just sits on one side, and we could even put one maybe right here as well. It sits on one side of it. These are called peripheral. Those are called peripheral proteins. Okay. So now what these proteins do is they can be used for anchoring. They can be used for recognition. So when we say recognition, all cells in your body have this little protein, it's usually a glycoprotein on it, and that says, I belong here. This cell belongs here, and so your body won't attack it. Okay, the proteins can be enzymes, and they can be used for transport. Okay, typically, if you have an integral protein, I'm not going to say all the time, but typically, it is usually a transport protein. Okay, an integral protein, and it's usually got a channel inside of it, and it allows things to go in and out of the cell itself. Okay. Another thing that we find that we have inside of the plasma membrane is cholesterol. Okay, and cholesterol. So what we're going to do is we're going to draw a couple more lipids, phospholipids on this side. And we're going to draw, let's just say a blue. We're just going to say we got this weird blue diamond shape, kind of weird thing. And we're going to say this is cholesterol. And we're going to draw some more lipids over here. Okay? And what the cholesterol does is it adds fluidity. It keeps the membrane fluid. Okay, that's what the role of cholesterol is. That's what the role of cholesterol is in the plasma membrane. So one thing I don't want you to think about is I don't want you to think about this plasma membrane as static, where the things are lined up together and they never move. The molecules, these phospholipids and these proteins and the cholesterol, they actually flow. They actually flow along the course of the plasma membrane, back and forth and all the way around the cell. And what it does is it gives it the name the fluid mosaic model, because it's kind of like a fluid structure itself. Okay, so now I want to talk about the transport proteins, because those are actually the most important part of this, in my opinion itself. So the transport proteins, okay, they can be receptors. They can be receptors. So we're going to make a new little line right here, transport. Okay, they can be receptors. So they have, a lot of them have, so they can't be. A lot of them have these things called receptors. Okay, and what the receptors do is they will bind and react to ligands, or ligands, whatever you want to call it. Now, this is important. So I'm going to use some words, some big words, some sciencey words. So a receptor is this. Let's just draw it out and explain it. Okay, let's just say I have a protein. Let's just say we're just going to call this thing a protein. Okay, and let's just say the top of this protein and the bottom of this protein, there's a hole in it. Let's just do it like that. Okay, so let's do it like this instead. Okay, let's say this protein is shaped like a hole. Okay, like a tunnel. And at the top of this protein, there is a door that is normally closed. Okay, so we're going to say the job of this protein is to open and close the doors. Okay, that's what we're going to say. And if the doors are open, stuff, and it doesn't matter what it is, stuff will pass through if the doors are open. But if the doors are closed, stuff can't pass through. Okay, now we have to find some way for the protein to know when to be open and when to be closed. And that's what a receptor is. On these proteins, what you do is you have this little part of the protein that's got a distinct shape. Okay, and let's say in this instance, stuff is shaped like a square. Now, when stuff bumbles around, and stuff is going to be moving in any direction whatsoever, and some of that stuff is going to get stuck in, let me change that color. Some of this stuff is going to get stuck in that spot on the protein. That spot is called a receptor. Okay, and when stuff activates a receptor, it is called a ligand. Okay, this is a ligand. And a ligand is something that will bind to and activate or turn off a receptor itself. Okay, so now when I bind this receptor, when this ligand goes into this receptor, what is going to happen is this protein is actually going to change shape a little bit. And what it's going, oh, I didn't want to erase that. And what it's going to do is it's going to open up the doors, and now stuff can go through the protein and get into the cell or do whatever the protein needed to do. Okay, now I don't want you to think that it's just for transport. Okay, there's this thing that I want you to start thinking about early, and I want you to start getting it in your head. It is called receptor mediated. Receptor mediated. The easy way to say this is your body, your cells, will not do anything unless they're told to. Okay? So now there are two ways to think about it. This is actually brilliant, or this is the dumbest thing I've ever heard. Okay? It depends on the day, the week, how I think about it. Okay? So let's just say, for instance, that I need to conduct electricity. I need to move. You're not going to move unless you get an electric current going. So how do we generate an electric current? Well, we have to open up channels. Well, what tells it to open up channels? Electric current tells it to open up channels. Okay. So if your cells want to divide, do your cells just know to divide by themselves? No, they don't know to divide by themselves. There is a signal that says, hey cell, it is time to divide. It is time for you to make a copy of yourself. Okay. When your immune system starts fighting infections, do you think your immune system just knows, hey, it's flu season, let's buck up our immune system so we can fight the flu easier? Nope. It has to see the flu and it has to be told to fight the flu. Okay. How does it do this? It is receptors. Receptors do this. Okay. One of the best examples of this is every one of you at one time has taken an antibiotic. So now it doesn't matter what it's for. Suppose you got an infected cut. Suppose you had tonsillitis. Suppose you had like a GI thing where you had like some horrible diarrhea and you had to take an antibiotic for that. Oh, and so when you take that antibiotic, you take it orally. Does the antibiotic know where to go? And the answer is no. What the antibiotic is going to do is going to go through the GI system. It's going to be dissolved into the blood. Okay. And it's going to be carried into the blood. And what it's going to do is it's going to bumble its way around in your body until it knows where to work. And how does it do this? It binds to something. That's how it works. It binds. And that is called receptor mediated. Anytime you want to get something done in your body, you have to have a receptor and you have to have a ligand bind to it. And what that does is it will tell the cells or it'll tell some other system to do work. Okay? So I'm done with that right now. We'll see more of this as we go. Okay, so the carrier proteins, going back to transport proteins, then we have these things called carrier proteins. Okay, and their name is self-explanatory. What they do is they transport things through the membrane. Solutes. Let's use the word solutes through the membrane. Through the membrane. Okay. And then we have these things called channels. Okay. And they allow water and other solutes to flow through. Flow through the membrane themselves. Okay, so those are our transport proteins. Okay, there are also carbohydrates of the plasma membrane. Carbohydrates of the plasma membrane, and they form what's called the glycocalyx. The glycocalyx. And basically what this is a sticky sugar coat. It is a sticky sugar coat. Okay, that covers all cells. All cells have a glycocalyx itself. Okay, and it's made from special sugars called proteoglycans, and you've seen this before. Proteoglycans, glycoproteins, and in some case, glycolipids. Okay. And what it does is the functions of the glycocalyx. is lubrication and protection of the cell. The cell itself. Anchoring of the cell. And locomotion. So movement, locomotion itself. And binding specificity. So receptor function. So if you want to draw a diagram, so let's just say we have our cell right here. Let's say we have our cell. Okay, we have our cell. We have our nucleus. Okay. And inside of the cell, we have some proteins sticking out. These are our channels, and these are, yeah. And on the inside, we have these peripheral proteins all scattered around. And then outside of all of that, we're going to do it in a nice orange. Every cell has this sticky layer called the glycocalyx, and it surrounds every single one of our cells. Okay, so it's not phospholipid bilayer. Okay, it's not a phospholipid bilayer. It's kind of it's a protein sugar or protein lipid mix itself, and what it does is it allows cells, to stick together, to move across each other. So let's say you have another cell right here, another cell right here. Let's draw a nucleus. And we got some protein sticking through this cell. We're going to have some integral proteins here. And on the outside of this, we're going to have the glycocalyx. And so they might even share a part of the glycocalyx on the outside. So that's what it is. It's just present. It's just present. Is it going to ruin your anatomy knowledge if you don't remember that we have a glycocalyx? No, definitely not. It's one of those things that you can treat like an empty beer can, and when you're done with it, just toss it out your shoulder, because honestly you're never going to use it again in your professional careers unless you become a microbiologist. Then it might be important, but that's it. That's what we find, the glycocalyx. All right, so now we're going to talk about some projections of the plasma membrane. So plasma membranes are not flat. They're not these little flat things. It would be awesome if they were, but that's really not how it works. Projections. Okay, and so the way to think about projections is just things that stick off. Things that stick off. And you have two that I really want you to remember. Two that you really want to remember. We have the microvilli. The microvilli themselves. And then we have cilia. Cilia. Okay, so microvilli are just finger-like projections. Finger-like projections. Okay, and their purpose is to increase surface area. So up arrow for increase surface area. Okay, now if you don't remember what surface area is, okay, so you can take a sheet of paper, or take any kind of paper itself. And so let's say this is 10 inches and this is 8 inches. So in. So area is just length times width. So the surface area of one side of this is going to be 80 inches squared. Okay, but we've got two sides to this. We got two sides to this times 2. So that's 160 inches squared. Okay. Now let's just say that I take this. Let's say that you take this piece of paper and you crumple it into a ball. Okay. So you get angry. You're like, damn note taking, and you just crumple it into the ball. Okay. What have you done to the surface area when you crumple it into the ball? Okay. Now a lot of people will mistakenly think, well, I've decreased the surface area. I'm like, well, really, have you? Have you actually decreased the surface area? And the answer is no. No. By putting all these little crinkles and stuff like that and all these folds in it, I can decrease the volume the volume that this thing takes up, but I don't decrease the surface area. And that's what microvilli do. So I'm going to stop drawing the plasma membrane, individual molecules, and we're just going to start drawing as a straight line. So basically, if you have a cell over here, and if you start having these little finger-like projections, okay, what I've done is I've got all of this plasma membrane going up, and I've got all this plasma membrane going down. I've got all the plasma membrane going up, all the plasma membrane going down, all the plasma membrane going up, all the plasma membrane going down through each one of these. So I am increasing the surface area. Now compare that. Suppose that I just had this. Okay. Now suppose that was the actual cell itself. There's a lot less surface area there. By putting these little finger-like projections, I increase the surface area of the cell. Now you're like, well, who the hell cares? I mean, why do you even give a damn about this? Because you're going to see when we start talking about the GI tract, or any part of the body that actually does absorption, where it needs to take stuff from the extracellular fluid and bring it in, you're going to want a massive amount of surface area, because you only have a limited time when as things are flowing through the small intestine and through the colon and stuff like that. A very limited time to get material from the fluid inside of the lumen into the bloodstream. So maximize that time by increasing the surface area. And that's what microvilli do. And microvilli, they attach directly. Let me put it in red. Attach directly to the cytoskeleton. And you're like, you haven't talked about the cytoskeleton. And I'm like, 100% correct. I have not talked about the cytoskeleton. I will talk about it. But basically the cytoskeleton basically gives the cell its shape. It keeps the cell together. So I'm going to do the cytoskeleton in red since I wrote it in red. And there's a cytoskeletal element sitting, and it holds a plasma membrane onto the cell. And there's these little cytoskeleton parts that go up through each microvilli. And each microvilli actually is anchored to the cytoskeleton. So it's an actual anchoring, the cytoskeleton of the microvilli. Okay, so is that important? Nope. I just put it in there because I just did. It's more important to be able to recognize them if I show you a picture of them, and I'll show you pictures of them in lab, and know that they just increase absorption. They increase the surface area. They don't move. There's no movement. Okay, so now moving on to cilia. Okay, now the cilia are hair-like extensions. Actually, I'm going to bring this down a little bit. Cilia are hair-like extension that move substances across the cell membrane. Okay, and that's what cilia do. So what you have on this one, so you have a cell. And you've got these long extensions, and they actually move. They actually move backwards and forwards. So these are the motile. They are motile. Okay. Now you're like, well, what are these for? Well, several reasons, and probably most important is clearing out the gunk that can gather up on top of cells. You find these in a lot of places. You find them like in the ovaries. Okay, so like in the ovaries of the female. Okay, got these fallopian tubes here that connect to the uterus. Uterus itself. Oops, that's terrible. That's the worst drawing I've ever done. And I've done some pretty bad drawings. Yeah. So let's just say you have the uterus right here. And on top of this uterus, sorry, not on top, but on the side of the uterus, you have the fallopian tubes, and they're the ovaries. Now the ovaries and the fallopian tubes really don't touch. And so let's just take a bigger picture here. So here's the ovary, and the fallopian tube sits right here like this. And so when a woman ovulates, it's kind of like, you got the little egg, you got the egg sitting right here, and the egg kind of takes like a leap of faith. It's kind of like base jumping, and it just pops out. It's pushed out, and on these, let me draw it in a different color. On these cells of the fallopian tube, there are these cilia. The cilia, and what they do is they kind of like grab it and push it along. They push it along to the uterus to be fertilized. Okay, so if you didn't have them, if you didn't have these cilia, there would be a distinct chance that the egg would be like, whoa, and then gone forever. So we need the cilia. You also find cilia in the ductus deferens of the male ejaculatory duct. They propel the semen or the sperm, up through the ejaculatory duct to be mixed with the prostate. Another place you find cilia, is in the trachea. Okay, and I think this is by far the most important thing, because every time you breathe in, you're breathing in fecal material, you're breathing in dust, you're breathing bacteria, you're breathing in fungi. If you're a smoker, okay, I'm not judging, I used to be a smoker. What you do, you're inhaling all those particulate matter. And what the cilia and the trachea's job is to get that gunk out. It's to actually propel it upwards. So whenever you have that, you feel it, you bring it up, and then what you do is you swallow it. You swallow it down and you get rid of it through the GI tract. So cilia, that's what their job is to move things along the surfaces of cells. Okay, so basically, cilia are typically smaller and thinner than microvilli. Microvilli don't move. Microvilli increase surface area for absorption, and cilia are there to just move things along. And again, I will show you this stuff when we actually get in lab. We have models of cells, and I have some pictures of cells that I will show you when we're going through this. Yeah, makes sense. Okay. All right. And so with that, I think I'm going to end it there. We're at a half hour. Damn it. I'm sorry. Half hour. I'm trying to keep them low, but shorter, but I jibber jab. So what we're going to do in the next one is we'll talk about membrane transport. So you remember that list in the beginning where I said the shit really doesn't belong together. So we're going to start sorting that stuff out, and we'll talk about how we get things inside of this, from the outside of the cell to the inside of the cell, and from the inside of the cell to the outside of the cell. All right.